My name is Jonathan Pawson, and I pastor this congregation right here. So let me first and foremost tell you that I'm very pleased to see you all here tonight. And my desire, as well as all of our desire, is that your time with us will be a pleasant one. And so if you have any questions, comments, or concerns that you have, please let myself know. And we will do whatever it possibly takes to help you improve or enhance your time with us here at these meetings. Another note, if you have a child that is 8 and under, we are offering some child care. And so it's off in the back. So I just wanted, that was a plug-in at the last minute here. Friends, we're really in for an exciting seminar. This topic, or this series of seminars or lectures that we're having, is dealing with the topic intelligent design or evolution. This specific field of thought has really piqued an interest in the scientific world. And a lot of, many scientists are looking at this. And I'm sure that this is going to be an interesting and exciting journey that we're all going to go on here during these series of lectures. But before I get going here, I have the privilege of introducing our speaker. His name is Jim Brackett. He is a retired physicist and a science teacher with over 40 years of experience in this field. He has lectured at various speaking engagements worldwide. He also has a passion for healthy living, and he's co-authored a book entitled 7 Secrets Cookbook with his wife, Neva. Jim, we're just very, very pleased to have you here with us. And we are looking forward to going on this journey and seeing this intelligent design or evolution discussion. So please, welcome.
That was some meal, wasn't it? I was sure tempted to put my hand up when she said, "Who would like a second dessert?" I won't ask you if you did that, but it's nice to be here. Some of you know that one of the special things about Bend is that my sister lives here, and her, my beloved friend, her husband Bob. So whenever we travel, if we can possibly pass through Bend, we try to do that. Sometimes we don't even give them warning. We just call them up and say we're in town. Now, when my youngest daughter was growing up, she loved to play baseball. And if it wasn't raining and if it wasn't dark when I got home from work, she would get her bat and her glove and her ball and say, "Daddy, let's go play baseball." So now she's a mother, and this little boy, he's a little older than this now, but he couldn't even walk yet. He could just stand up, but she wanted to teach him early. And here he is. This is the wind-up, and this is the pitch. I knew you'd be disappointed if I didn't show you that picture. In any case, there's some big questions that thinking people always face in their lives. Where did I come from? Where did we come from? Is there some reason why I am here? Why we are here? And finally, where am I going right now in life? What am I supposed to be doing? And perhaps, for those who are thinking about the possibility, what am I going to do after that? And I'm not really going to deal directly with the second and third questions. Our lecture series is essentially on the first one. How did we get here? There's really only 2 in somewhat broad classes in a way, and that is that we must have evolved because here we are, or many people, as you know, there was a creator or a designer at least, and most people who have adopted intelligent design or that concept are people who think that there is a designer who is God. A few people, some notable people, some very bright scientists, they say and we'll get into this more in a moment, they say, well, I don't know. I'm pretty sure there was a designer. That's pretty obvious to me, but I don't know more than that, which is an interesting place to be. And where did we come from? So was it evolutionary processes? And we're going to look at this more closely, of course, each evening and even this evening. But, and I realize that we have a wide variety of people here in terms of background, knowledge, experience, training, education, which is an interesting challenge. I spent most of my life teaching science, and usually the whole class comes in, they're kind of in the same place. They're just starting to take whatever, whether it's Algebra 1 or whether it's physics or something. But we have people in here across the spectrum, and I'm always interested in how to try to relate to that. So some of you know very much about this, and some of you aren't maybe too clear. But the evolutionary process, if you could describe it, is that there are mutations by accident in the genes that are in our cells. And that those mutations, and that's a word I want you to learn. There's a number of things I really need you to just grab onto, because you know what it's like if somebody starts using terms you're not familiar with, and then they start using other terms that you're not familiar with that depend on that term. Pretty soon you just feel like it's lost. So I'm going to try to point out to you some things that I would like you to really hang on to. And if we should have supplied some paper for you to take some notes. I would like you to know what this word mutation means. And I guess I'm standing too far out. Is that the problem? Okay. It didn't happen the other day, but nevertheless. Mutation, the definition is a change in your DNA. And as you'll see in a minute, most of our DNA is not genes. I think most of you know that our DNA is where our genes are. We have only learned recently that most of the DNA is not genes. So it could be a change in a place that's not a gene, or it could be a change in the gene. And you'll understand more of that in just a few minutes. That's what mutation means. And in the evolutionary world, these changes are accidental. And there are many sources. This is established scientific fact that there are things that change our DNA. One of them you're very familiar with is radiation. Whether it was from an X-ray, or whether it was from a cosmic ray in the sky, or some radioactive material in the ground. Those things cause changes in our DNA. They can make us sick. The evolutionary paradigm is that if there was an accidental change in the DNA, perhaps in some gene, that would make the creature better, have some kind of an advantage over the creature's peers, whether this is a person or an animal, then that person might survive and the others might not survive. Because maybe this creature that had an advantage because of the genetic change could catch the prey quicker or easier. You get the idea? And I appreciate those of you that nod when I ask you a question. Now, I'm an old teacher. I'm not really a lecturer. And if you've ever been a teacher, so how many of you have been a teacher in your life sometime? There's a dozen of you. It's frustrating to me if you look at me while I'm talking and there's never anything on your face that says, I understand. So let me know. And by the way, Pastor, I did suggest for someone to buy some 3x5 cards, and we could pass these out if there are questions. My preference is to take questions live, but we would be here all night. And so I'm very comfortable with that. I would love that, but we're going to try to keep it down to this section of time each evening. And so what I've got to do is take your questions on paper and try to pick the ones out each evening from now on. Tomorrow night, I'll take a few minutes at the beginning to try to deal with these questions. And another thing is, if you don't write the question down when you're thinking about it, if you're like me, I forget the question I had. Does that happen to you? So if we can get these cards in people's hands, I see she has them. Just pass them out at the aisles. And if you don't have a writing instrument, borrow one from your neighbor. But I'm not just teasing, folks. If I'm looking at you and it looks like you're not letting me know it's clear, I'll think I didn't explain it well enough, and I'll try to explain it again. And you might get tired of that. So either nod if you understand, or even if you don't understand, just nod, and you'll fake me out. The idea with evolution is there's an accidental change in the gene or in the DNA somewhere else, which would give that organism an advantage over its peers. And then if that happened and the peers died off and that one organism didn't, then that organism's progeny would maybe, and there's some issues there, maybe pass that on to their offspring. In fact, that doesn't happen very often. See if you can follow this. If some DNA somewhere in one of my cells got changed, that doesn't pass on to my progeny. It only passes on to my progeny if I'm a male, if the sperm got changed. Are y'all with me on that? Or if it's a female, if one of her eggs got changed. Are y'all with me on that? It's an interesting question. It's a very special place to make the change, see, to pass it on to the progeny. But that's the theory behind the evolutionary paradigm. And there is no question that these changes do take place by radiation, by toxins. There are things that we have in the atmosphere today, maybe more than we used to be, but they can cause toxins. Now, this is something no animal would ever do, but there are over 7,000 poisons in cigarette smoke, many of which cause mutations in our DNA. So there's a number of things which cause mutations, and the scientists who believe in the evolutionary paradigm see that as the mechanism. That's the Darwinian mechanism for changing an organism, whether it's a cell, a single-celled organism, or an animal, and everything in between, that would happen. And of course, on the other side of this picture, these are about the only two possibilities that people come up with. We were designed, animals were designed, plants were designed, and there must be a designer if they were designed. And most people, as you all know, most people, not everybody, say, well, that designer, that would be a god, and this designer would have the ability to make what he designed. Well, how would we get the answers to these two questions? I don't know if you're aware of this, friends. We cannot prove evolution or design creation using science. We can't prove it. By definition, the scientific method means you have to be able to take this thing into the laboratory, if you will, and make it happen. Replicate it, see. So both camps are an interesting place. The people in the evolutionary paradigm are absolutely, they believe that this has happened, that this is how we got here. And the people in the creation or design camp are sure. And that's interesting to me as a scientist. And I'm in one of those camps, as you all know. And I believe it's fact, but I can't go into the scientific laboratory and prove it. Y'all with me on that? And I'll give you an example of that tomorrow night, just to make sure we're kind of all on the same page. So even though Robert Dawkins, who is the current, perhaps most well-known evolutionist in the world, says that evolution is a fact, and he doesn't. Now, there's something else that we got to think about here. That statement, or even the idea of evolution, you got to be careful what you mean. You have to know what you mean, because we have clear evidence that a single organism, a little germ, a little microorganism, gets mutated, and whether it's radiation, and we do this in the laboratory. We can prove this in the laboratory. Irradiate that little microorganism with X radiation or radioactive radiation, and you can make a new species of organism. Y'all with me on that? You can do that rather easily. You could have high school kids do this with some pretty simple equipment. Now, making sure it's a completely different species is a little more of a challenge. You got to have some microbiology skills in that, but that can be done. What we have not proved is that if you had a given, we use a fancy word for it, phylum, a creature in some class or some kind. We haven't proved that you can make some mutations and make that, watch this, make that dinosaur into a bird. I don't know if you know this, but the evolutionary community believes that birds came from dinosaurs. And there are many other pathways that they feel are the way that these creatures formed. And we cannot prove that. We cannot go to the laboratory and make that happen. So we can't prove that kind of evolution. And we tend to call that macroevolution. And then this business of changing a microorganism, do you know what I mean when I do this? I'm talking about something you can't see but with a microscope, like a germ. And not all microorganisms are germs. They're not pathogenic. They don't make us sick. We know that you can make a different species, but we've never been able to take a microorganism and make it into a muscle cell, right? We can't. We've not been able to do that. So, in spite of the fact that Richard Dawkins says evolution is a fact, it's just as much a fact as the sun going around the earth. That's just not true. We cannot prove that. On the other hand, the person who believes in creation, we can't go to the laboratory and say, where's the designer, please come in here and make something. We don't know how to do that. You understand that, don't you? So what kind of evidence? What are we going to use? Well, in the world of design, which I'm going to spend some time on this evening, we look at what we know about living things, and most all of you are very aware of this. It has become astonishing, the complexity that's here. Tomorrow night, our interview is going to be with a PhD in molecular biology named Michael Behe. Most of you don't know that name. How many have ever heard of the book Darwin's Black Box? Can I see your hands? Well, I'm pleased to see that. Maybe 15 to 20 people. Michael, let me back up and tell you that the person who is the father of what we call intelligent design, and you all know that term, don't you? Whether you're in the evolutionary, most of you are nodding, but remember, folks, when I ask you a question, please let me know, all right? You know that term. We're familiar with that. The father of intelligent design is an attorney from Berkeley named Philip Johnson. Philip Johnson wrote several books, one of them called Darwin on Trial. Philip Johnson is a very astute attorney. His material is fascinating to read. I've read all his books. Never met him personally. I've heard him lecture on many occasions through media. But he's the father of the movement. But he was inspired by an agnostic, actually an atheist, and we'll tell you this story later, who wrote a book called, I got to think of the name of this book, Evolution, a Theory in Crisis. This man is an evolutionist. He was an atheist, and he saw problems with the evolutionary theory, and he simply categorized this in a book. This is fascinating reading. I'll read you a few paragraphs from it. Not tonight. Fascinating reading. And Philip Johnson read this book. You'll hear this story. At the same time, he read Richard Dawkins' book, The Blind Watchmaker, which is about the evolutionary paradigm. And here were two, in his mind, here are two esteemed scientists, qualified, both of them, credentialed scientists, saying the exact opposite things, looking at the same data. Very interesting. And so Philip Johnson started reading and thinking. He was very impressed, particularly with the book by Michael Denton, Evolution of Theory and Crisis. I'm sorry I've given you so many names. But Philip Johnson then began to publish and write and lecture, and he became the father of the intelligent design movement. But Michael Behe, through the book Darwin's Black Box, is what really put intelligent design on the radar screen of this country and the world. Are you all with me on this? So it's particularly why I wanted to interview him, and we'll have that tomorrow evening, a 1.5 hour interview with him, and he tells some very interesting stories about his own effort to understand. Well, the evolutionary community then are typically atheists or agnostics. Now watch this interesting point, friends, and assume that there is no supernatural influence or power. In fact, this has become the assumption of science. Because if you have to go into the laboratory and replicate something to call it scientific, you with me? And you don't have a God walk in there with you and go and make things happen. You're still with me? By definition, science says there is no supernatural. All right? By definition. It's assumed. And the evolutionary community are scientists, most of them. I mean, there's lots of lay people that believe this too, of course. Now, interestingly enough, there are many scientists who believe in design and in creation. But they all recognize that they cannot prove that with science, to repeat myself. See, they understand that. All right. Let me just quickly tell you, you're probably aware of this. The word atheist comes from the Greek word theos, which is the name God. You translate that in Greek as God. And atheist is someone who says there is no God. That's an interesting position. A little bit arrogant, in fact, if there happens to be a God out there, for me to say there is no God. Now, an agnostic, I really think that people ought to take this position. That comes from the Greek word gnosko, which means to know. And so to know, or agnostic or agnostic means I don't know. Y'all with me? An agnostic says, I have no knowledge of this. I don't know. I think that's a more defensive posture. So, this is just what I've said. It's widely believed that scientists are, now notice this, it's widely believed that scientists are virtually all atheists or agnostics. That happens to not be the case. And in the book, Science Discovers God by Ariel Roth, he quotes a survey that was done recently that shows that 40% of scientists in this country believe in a prayer-answering God. Isn't that interesting? That's very interesting. Now, they go back to work the next day, and there they are in the university where everybody is just about everybody, almost without exception, is an evolutionist or believe in the evolutionary paradigm. And so they're kind of in that world. And here's what has come of this. There's this class which is kind of a third category. We've talked about the evolutionary paradigm. We've talked about the creation, designer paradigm. There's actually a class in a way in between that says it's, in fact, the name of it is theistic evolution. Now, you know about that, probably. It means that, well, there's all kinds of different theistic evolutionary ideas. But the basic idea is God started it, and then he let evolution make all the different creatures. Some people think that he maybe came in and helped it once in a while, and so forth. So that's why there are different ideas about that. The Catholic Church, the largest Christian organization in the world, teaches theistic evolution. They believe that God started it, and then let evolution make all the creatures that we see. Now, the people who are strictly in the evolutionary paradigm have this problem. Where did the first life come from? And I'll have to say that there's just no clue for that in the evolutionary paradigm. Absolutely no clue whatsoever. But you say to yourself, if you're in that world, well, it's here, so it had to have happened. I don't know how it happened, but somehow the first cell came to be. And one of the problems is that the evidence we have from fossils, and I know you all know about fossils, but just to be together on this, a fossil by definition is some evidence of a living creature, that the living creature there may not be any of those alive anymore. Have you heard of a living fossil? Yes, yeah, the term is used. And what it means is, we thought that this thing, that species was extinct thousands and millions of years ago. But we suddenly find one, and so we say, well, it's a living fossil. But in any case, the definition is some evidence, and often you know about this, it's an imprint of a leaf in mud that turned into a rock. So someday I go out there and break this rock open, and here's this imprint of a leaf, or of a fish, or of an organism of some kind. And the problem is, according to the evolutionary geologic timescale, 580 million years ago, there was no life on this earth. And I say we, the scientific community, you all know something about this, has figured out that there are rocks that were made not long ago maybe, but rocks that were made longer ago than that, and you keep going down this so-called geologic column. There's no place in the world where you see the whole geologic column all in one piece. That doesn't happen. But we pretend like it's there to study the idea, and that's okay. We take it from different places, and we say, oh, this place only has so much of the geologic column, and over here there's some of it, and so we kind of put it all together. And you come to a place down there, if you get deep enough into the geologic column, far enough years back, there's no life. There's no fossils. No evidence of fossils. And even though it may sound like a long time to you, over a period of about 10 million years, that's what we think was how much time went on there. I say we. This is not my own opinion. I'm talking about the scientific community in general. All of a sudden, you see life in a very complex form. You don't see life, you don't see simple life. It's instantly complex. Now, there is a portion of the early part of that Cambrian explosion. You've heard that term, haven't you? How many have heard the term Cambrian explosion? Like 90% of you, 80% of you. In the lower part of that, there are some single-celled organisms. But very quickly, you suddenly find these complex organisms. There's no such thing as a simple organism that existed first. Even these single-cell organisms are complex things. So we just don't know. We don't know how it all started. It's just suddenly there. And if I'm an evolutionist, I just have to say, well, I don't know, but there it is. So it had to happen. It had to have happened. And then, of course, I've got the problem of saying, how did the Darwinian mechanism make all the other creatures? Which is a huge problem, as you probably already know, and we'll look at some of that. I'd like to say at this point, folks, that even though I'm from the creation world in my own philosophical position, it's always bothered me that people who are creationists make fun of the evolutionists by saying something like, well, you might have descended from an ape, but I didn't, etc. I don't think that has always bothered me. And I thought for many years that the evolutionists were above that. That's not the case. They make fun of the creationists too. And so we all ought to quit doing that. Don't you think? There are very smart people who believe in evolution. Very smart people. There are very smart people who believe there's a God and it was created. Very smart people. So it's an interesting world, isn't it? Well, here's an interesting story. Before Richard Dawkins, what was there? There was a man named Antony Flew who just passed away 3 years ago. Antony Flew was the most respected atheist for 50 years in this world. He wrote, by the way, he was the son of a pastor, became an atheist as a young boy, and wrote many books on many topics, and was best known, however, for his arguments that there was no God. That's what he was best known for. Debating people and saying there's no God and giving reasons for it. But in 2004, he began to change his mind. This is the world's what? Most respected, most famous atheist. And his last book was titled, There Is No God. And the no was scratched out. And what was in its place? There is a God. And the subtitle is How the World's Most Notorious Atheist Changed His Mind. And on the book cover, behind the title type, is a bust of himself upside down. Can you see it? What's it saying? I've been turned upside down in my thinking. Are y'all with me? And it is wow. And this is a little press release in 2004. Here's a comment from another newspaper about this British philosophy professor, a leading champion of atheism for more than half a century, changed his mind. He now believes in God. Now, here's an interview with Antony Flew, and because he's a Britisher and has a little bit of an accent, I'm going to put the words on the screen as he says them, so you can kind of stay with it. I'll actually put on the screen the words of the interviewer as well.
What were some of the factors that prompted you to, in recent years, reconsider atheism and to come to the conclusion that there is a intelligence?
It's been entirely these biological discoveries, the integrated complexity argument. If the integrated complexity of the physical world is a good reason, as Einstein clearly thought it was, of believing that there was an intelligence behind it, then this argument applies a fortiori with the inordinately greater integrated complexity of the living world. It seems to me this is just obvious that that argument is much stronger now.
Fascinating. Fascinating. Now, I could also find people, I have some friends, who were once believers in creation design, who have become atheists or agnostics. So this is no proof, is it? Are y'all with me on this? This is not a proof. It's very interesting that there are many leading scientists, and we will interview a couple of them, who were once evolutionists and have decided, because of, what's the term? The inordinately greater integrated complexity of living creatures, that say it is far too complex. And one of my efforts tonight and in the preceding evenings is to try to carry you a ways into that world. I was doing this lecture in Seattle a while back, and my wife's brother was there. Now, he's an intelligent guy, just a really great guy. And he said, well, it all went over my head. Well, that really bothered this old teacher. Are y'all with me on that? So I need your help this evening and all the evenings. There's going to be there's going to be the possibility that you will say to yourself, now maybe nobody would quite do this, I don't do science. In fact, there was a lady, Tom, at our lifestyle center about a year and a half ago, maybe 2 years. She's a teacher, taught elementary school all her life, and she said to me, I don't do science. I don't understand science. And that just, and she was good for her word too. And in our lifestyle program, I teach nutrition. That's science. And some of it's pretty deep, is that right? Some of it's pretty deep, but she wouldn't brook that. But I want you to promise me tonight. Raise your right hand, please. I promise I will do my best to understand. Integrated complexity. See, that's more than just complexity. It's like this terribly complex thing would never work without this terribly complex thing somehow cooperating with it. Integrated complexity. And you and I, in this series of lectures, are going to just plunge in fairly shallow. But many of you, if you don't really work with me, are going to get lost. And so I really need your cooperation. You've all promised. Just keep that in mind. So in order to even look at the possibility of an evolutionary pathway, or to grasp why many scientists believe that life was the result of intelligent design. In both of these cases, friends, wherever you might be in your own personal philosophical place, you've got to have some understanding of this thing we call DNA. Are you ready? All right, here we go. We're going to need to have a knowledge, what it is, what it's for, and what the consequences of those things are. Now, here's a Newsweek magazine back in '04 where they put a picture of a piece of DNA with grandma on one side and granddaughter on the other. And the reason they look a lot alike is because they have similar DNA. How many of you before this evening would be able to say, I am aware that DNA is kind of like a ladder that you twist in opposite directions and you get these things they call. How many would know that? Can I see your hands? Now, all of you know it now. Are y'all with me? DNA is a ladder. Okay? And the ladder has rungs. You can see that in the picture, can't you? And it has a lot of rungs. In fact, I think I have the number on the screen. There's another picture of DNA. I like this one because the rungs are in two colors. Can you see that? There's a reason for that. We'll come to in a minute. Here it is again with colors. And what it's showing, this is what it's showing. A rung of a ladder you buy is one piece, but the rungs in DNA are two half rungs kind of hooked together. How many would have known that? Can I see your hands? Not many, but you know it now. Half rungs. Are y'all with me? Half rungs. And if you look carefully at these colors up there, the artist has correctly colored the half rungs with only 4 colors. Can you look at that and see that? What he's trying to say, or she, whoever drew this, is that scientists back in 1953, when they figured out what the DNA was shaped like and how it was made, figured out that these rungs only came, these half rungs only came in 4 different kinds. It's an interesting story. And most of you know, many of you know, Watson and Crick got the Nobel Prize in 1954 for the discovery they made in 1953 about this ladder with this double helix. One helix going like this and the other helix going like this, twisted in opposite directions. I like the one on the left the best of all the pictures I've shown you, because the artist has correctly shown that the half rungs are not really touching each other in the middle. I'd love to take a half an hour and tell you what's going on right there, but see, that would be too deep. Too deep. Since you said that, I'm going to tell you. It's the hydrogen bond. How many have ever heard of such a term? Good for you. It's the I made a mistake and didn't tell the folks that I need a whiteboard. Is there a whiteboard in the church that you guys could drag in here so that I could go write on it once in a while with some pens that work? You know how those are, and they almost never work unless they're brand new. I apologize about that. I'm going to fix you for saying that. We'll just take a minute on it. Anyway, this is a correct picture. Not terribly important for our discussion, but it's just interesting to know. And here's how many rungs there are. Some person under 15, tell me what that number is. What's that number? 3 billion. The number is actually 3,200,000,000. But, and if that was a regular ladder, where the steps were about a foot apart, that ladder would stretch from here to the moon and back and halfway back to the moon. That's a lot of steps, isn't it? If those steps were as close as paper sheets, it would still stretch from here to almost Seattle. That's a lot of steps in that ladder. It turns out the steps are 1,000,000 times closer than even paper. So that the whole ladder, y'all with me still? This is a ladder with how many rungs in it? Twisted. The whole ladder in your cells is actually about this tall. And I'll show you tomorrow night how the body wraps it up into a piece that is smaller than a microscope can see. Very amazing. And the sinus, you know, this didn't happen Saturday. What's going on? But I was way out here during the... Okay. I don't like to be up there. Scientists were amazed to discover just a few years ago, they thought that there should be well over 100,000 genes. There maybe should have been several 100,000 genes. And when the DNA was mapped, we'll come back to that in a minute, they found there was only about 20,000 genes. And this just blew them away. This is recent stuff, folks. And let me give you another word I want you to be familiar with all week long. The whole ladder is mostly not genes. Are y'all with me on this so far? It's mostly, in fact, 98% of the ladder is not genes. And so we have a word that scientists use. They call this whole ladder the genome. You've heard the term? I want you to know what that means. It means all the DNA, the genome, even though only a tiny portion of it is made up of genes. Between the genes. In fact, the gene on the ladder is a piece of the ladder. Sometimes a long piece and sometimes not so long. The average gene has about, I'll come back to that in a minute. We're going to use another way to talk about that. 98% of this ladder is not gene, and there's another word I would like you to be familiar with. We say it's non-coding. What the scientist means is, watch this point now, folks. I think that's the next thing on the slide. A gene is a pattern for a what? For a protein. And what we say is it codes for a protein. What does that mean? Well, you know what? We use code all the time. Every word we use is almost a code. So you say to somebody, hand me the umbrella. That word umbrella is a code for something, isn't it? That goes pop when you press the button and shields you from the rain. Isn't that right? It's code. And if a Russian walked in here that doesn't speak English and you said umbrella, they would have no clue about your code. Is that right? See, and the gene codes, and I'll show you more of this in just a moment, for a protein. So this is what we say. In the genome, in that ladder, the part that is not genes, we say that's the non-coding portion. Got it? You'll see that in magazines and stuff. All right. Now, a pattern for a house looks something like the house. Oops, got to get the right button here. Thank you, gentlemen. Okay, because I can't come down here like I'd like to, let's put it up front. Okay, figuring this out. Oh yes, that's there. So this is the blueprint. So there's the bathroom. You can see the tub. There's the kitchen. There's the stove and the sink, and there's the dining room, and there's the garage. There's the pattern looks. Watch this. This is an illustration, friends. The pattern looks something like the final result. Are you all with me? That is not the case with a gene. The gene pattern looks nothing like the final result, which is going to be a what? You didn't all get it, but you know what my answer was supposed to be. The gene is a pattern for what? A protein. All right? There's no exception to that. That's what genes are. They are a pattern for protein. And I'll show you how that happens in a bit. So if it's not a pattern, what is it? It's a code. And I just mentioned to you an example of this. In our DNA, the code comes from the chemicals making up the half rungs. Ooh, what's that? How many kinds of half rungs are there? 4. I didn't tell you this, but they're just 4 chemicals. There's only 4. Even though there's how many rungs? Over 3 billion? It's just repeating the same 4 in different patterns. And the way the scientists do this to be able to talk to each other is they take the first letter of the name of the chemical and they give that letter, they give that half rung that letter. So you won't even have to learn the names of the chemicals, but you do have to learn the names of the 4 letters. Are you all with me on that? Okay. And they are what? Those are just the names of 4 chemicals with the first letter, right? Okay. And so when the artist drew 4 colors, he was trying to give you the A color, the B color, the T color, the C color, the G color. Y'all with me on the idea? That's what they were trying to represent. So here it is again. And if you look carefully at this, how many different colors is the artist using? 4. Even if you don't look carefully, you know the answer, don't you? 4 different colors. And it turns out that if this is the A color, and that's the C, and that's the T color, and that's the G color. They're not colored. This is just for clarity. You understand? In your body, they're not colored. All right. Don't want to get confused here. Well, now here's the next thing that happened. The scientists decided that they wanted to map, if you will, the whole genome. Map. Now they use a different term, and I'd like you to learn this term. They sequenced the human genome. In other words, they gave the sequence of what? The letters. Are y'all with me on that? They figured out the sequence of the letters. How many letters are there going to be? No. I'm sorry, I didn't make the question clear. When they sequence the entire genome, how many letters will they end up with? 3,200,000,000 letters. Are y'all with me? All right. How many have I completely lost? Let me see your hands. We're going to charge you 3,200,000,000. No. How many should have raised their hands and didn't that I've lost? Okay. What did he say? Oh, they shouldn't lie. No, they're understanding. Let's make it clear, folks. If you're lost, we have a ladder that has 3 billion rungs. Are y'all with me on that? You've got that much. You're not lost for that. And that ladder represents our own genome or our own DNA, which, by the way, is in every cell of our body. An exact copy, except for mistakes, and there aren't too many of those, an exact copy in every cell with 2 exceptions. And from now on, when I say it's in every cell, you just understand that I know there are exceptions, and I just don't want to keep having to say it. Red blood cells do not have DNA in them. The machinery inside red blood cells have been almost completely removed, so that cell can just do one thing, and that's carry oxygen. And the other cells are, in females, the eggs only have half of the ladder. They've got 3 billion rungs, but they've only got the half side. Y'all with me on that? and the male sperm has the other half. It's a little more complicated than that, but that's pretty close. Here is the book. It was during the Bush administration when this was funded. It cost $1 billion and took 10 years to sequence the human genome. I wish more of you would have said, wow. I don't want you to say it mockingly. You say it genuinely. I still heard mocking, but...
That's cool.
I do want you to be impressed, friends. This was a serious effort. Now, I'm going to tell you a story about this man I shouldn't, but when the scientists figured out that most of the genome was not genes, it blew them away. And the head scientist of this whole project is the director of the National Institutes of Health. Most of you know that term, don't you? That's a government organization which supervises all the other national institutes. National Heart and Lung Association, National Cancer Institute, National Heart, and you understand, all of these research organizations. The head of that is a fellow named Francis Collins, and he's a scientist. And Tuesday night, when we have Dr. Carson's interview, he will tell a story about Francis Collins that I'm going to tell you briefly. I think it will help you follow all what Carson says. They're friends, not surprisingly. And Francis Collins was an evolutionist and not a believer in design or in a God. And he says to himself, because in his graduate work he was studying DNA, and he said to himself, this is far too complex to just have happened by chance. This is the #1 scientist in this country. Is this impressive? This is impressive. And he became a believer in design and actually a believer in God. Interestingly enough, he had the audacity to say to himself, the rest of this DNA. Now, you can actually be a believer in God and be an evolutionist. Is that possible? That would be a theistic evolutionist. Are y'all with me? And that's kind of where Collins is at. And he looked at this genome that was 98% not genes, and he said, the rest of this stuff is just junk. And he wrote a book called Junk DNA. Have you ever heard the term? Francis Collins is the author of the idea. Now, to his credit, and to the credit of the governmental agencies, if you will, if there could be such a thing, I shouldn't say it like that. They put up the money to hire 400 scientists. I say they. This was an effort by many countries around the world. And together they have hired 400 scientists from all over the world who are, see if I can make this work like I'd like to, who have been given the assignment to see if the junk DNA isn't junk. And it's the ENCODE project, which stands for the Encyclopedia of DNA Elements. And what that means is they're studying the non-coding region. Y'all with me? Of the DNA, of the genome. Are y'all with me? Is there anybody in here who is lost who says, I'm kind of getting it now? Please, somebody raise their hand. Good for you. All right, we're going to do it now. This is not in the notes. This is free. ENCODE. Water. All of you know that water is H2O. I think you all know that. If you didn't, you know it now. That means there's 2 hydrogens, and I'm going to do it wrong, 2 hydrogens hooked to an oxygen. Hydrogens are tiny in comparison to oxygen big. And if this is the way that water occurred in nature, water would boil away, don't hold me to this number, at some temperature below zero. So water wouldn't exist. Are you all with me on this? And there would be no life. Now, this is the way it ought to hook up. If you just think about the way the electrons and everything works, this is the way it ought to hook up. The way it actually hooks up is this. At a certain angle of about like that, this is where the hydrogen atoms are hooked up. And because the hydrogen atoms are positive, and because the oxygen atom is negative, this side, because the hydrogens are over here, balancing the minuses and exposing this side of the water molecule tends to be negatively charged, just like when you brush your hair with your comb and it goes, are you all with me? That's electricity, isn't it? And the comb, you may not know this, has got negative charges on it, and the hair has positive charges on it, and you all know that positive and negative electricity attract, don't you? You all know that. So what you have here is one side of this is more negative and one side of this is more positive. Does that make sense from what I've drawn? And because of that, water molecules tend to hang together because this side is positive and it pulls to that one. And this keeps water liquid clear up to 212 degrees Fahrenheit. Is this remarkable or not? It's absolutely astonishing. And what holds the half rungs together? The A and the T are held together by two hydrogen bonds. This is the hydrogen bond here because one side, we say it's polar. You talk about North Pole and South Pole. In the electronic world, we talk about polar, electrically positive or electrically negative, and because these things are polar, the two half rungs are held together by two hydrogen bonds, and the other rungs are held together by three hydrogen bonds. There you go, it's all your fault. Y'all with me? Pretty remarkable, wouldn't you say? If it wasn't for the hydrogen bond, we would be dead, bang, just like that. Astonishing. Absolutely astonishing. All right, so this is the book. And this is the scientist who's head of the 400. Very cool story. I can't go any further because of time. And here's the book. This is the same picture, friends. I just took a close-up of it. Y'all with me? What do you see in the book? A's and T's and G's and C's. And how many are in the book? 3,200,000,000. Cool. Now, you might say, so what does this matter to me? I'll tell you what it matters. It matters a lot to Angelina Jolie. Listen to this story. Every one of you in your DNA has, among many genes, you have two genes that when scientists first found them, figured out that these genes had something to do with breast cancer, so they named them BR for breast and CA for cancer, and they found one and they found another one. How many have ever heard of Brackett 1 and 2? Can you see your hands? And we learned after a time that it's not just breast cancer. I'll come back to that in a minute. But we all have these genes, and these genes, we have given them a name, tumor suppressor genes. Both of them are. They prevent us from getting cancer. And to begin with, we saw that they were preventing breast cancer. We now know they prevent cancer all over the body. And a gene, remember folks, a gene is a section of the ladder. Is that correct? Y'all with me on this? And it has to have the right A's and T's and G's and C's to make it work. Are y'all with me on that? And what happens is there is a mutation that can happen and change one of these letters to a different letter. And now this gene can no longer prevent the cancer. Still with me? And this, an average American woman has 1 chance in 8 of getting breast cancer during her lifetime. That's almost 13%. If a woman has this mutation, do you know the word? It means there was a change in the DNA. She inherited. This didn't happen because of radiation on the woman. Because if you change just one cell, the gene in one cell, it doesn't really make a lot of difference. Are y'all with me on that idea? She inherited this gene from her parents. So every cell in her body has a BRCA1 gene that's been mutated, which increases her chance of getting breast cancer to something above 80%. So you all know what she did, don't you? She had both her breasts removed. So was this a practical thing for Angelina? You're missing my point. If she wanted to protect herself from getting breast cancer, did the fact that there was a human genome that had been studied and figured out, was that a practical thing for her? Y'all with me on the idea? And today, if you want, I'm not recommending this necessarily. Did I put the pen back here? Today, if you want, you can get on the web. There's a place called 23andme.com. By the way, Google is the one that made this company. You may not know this. Google has made a bunch of companies. Some of them have failed. And this company will check your genome for, I think it's only $100. They will check particularly the tumor suppressor genes to see if they're okay or if they're mutated. Got it? And they're trying to see if they can't make some money, but they actually are altruistic and they would like to, help some people. So I understand what those of you who said no, it wasn't practical for her to do that. I'm not here to discuss whether or not she should have, but I want you to see that this is just a tiny example, folks, of the things that could come out of knowing what the human genome is, sequencing it, mapping it, if you will. Okay. So let's take the rest of our time this evening to kind of delve a little bit more into this world of integrated, what? Inordinate, integrated complexity. And every night I'm going to do a little bit with you on the DNA, and I'll review quickly what we did tonight, and I'll try to carry you along so that you'll have some facility to understand and be thinking about why evolutionists have their opinions. And some of you are in that camp. I realized that very few, it's interesting, we did that the first time I did this several years ago. I was requested to do it, I spent a year reading about 20 different books, even though my world is the science world. I wanted to try to get as informed as I could in this whole thing. I've lost my thought. There was this story about somebody who, oh yes. No, one of the young women that was in this church where I did the series was taking science in the local university, and she went to her biology teacher, and she gave him a copy of the flyer, and said, "Please come." And he said, "I'm interested, but I'm not interested." And I understand that. He is certain that intelligent design is foolishness. Think about this, friends. See, most of you are believers in God. Is that right? Some of you are not, which is, I'm glad that you're here. I respect very much your opinion. But you who believe that there is a God who has this power to create, or at least to start life and maybe watch it become all the different creatures. And if that's your opinion, that's okay. Put yourself in their position. Have they ever seen any direct evidence of divine intervention? Probably not. When I say direct evidence, I'm talking about scientific evidence, right? And would it be hard for them to imagine, as they study the cell and they look at the pieces of the cell with an electron microscope and they see how tiny this stuff is, they can't imagine somebody that could reach in there with their big fingers and even put this stuff together. Does that make sense? They just can't imagine that this is the case. And they are immersed in this community of people who believe that this is not the case. And so it's very easy to say, it just couldn't be. This is just crazy. This could never be. You understand the idea? Sure. So do you think we should be respectful and listen to them and have meaningful dialogue with them? Sure. I feel bad that he didn't come. But what he was saying to his student was, it would be a waste of my time. I know what the facts are. Kind of an arrogant thing to say. But nevertheless, for those of you that are in some part of that world, I'm just pleased. I'm delighted that you're here. I hope you just are willing to weigh evidence on both sides. This is a book in my library. It took, what is it, 8 PhDs? 1, 2, 3, 4, 5, 6, 7, 8 in their expertise to write one textbook on molecular cell biology. Now this is a graduate text, but this would be the first high-level graduate school. This is not the last book that they would have on molecular biology. And if you started reading this book, it would blow you away on about page 2. Integrated complexity. Y'all with me on this? But we're going to try to just get some of it, just so we have just a sense of what's going on. Inside that book, many pictures of cells. And the artist has cut the top off of this cell. There's the nucleus. He cut a corner out of the nucleus. This is where the DNA, this is where the genome material is. It's in those little squiggly lines in there. Those are the ladder pieces. You probably know that the genome is not in one long ladder, even though I've pretended like it was. It's in 23 chromosomes. Are you with me? You all know the word chromosomes. How many people knew before they came here tonight that that's where our genes are? Can I see your hands? See, not every hand is raised. But you all know now. Is that correct? The chromosomes are the DNA, right? So that ladder that's 3,200,000,000 steps long is made up of all those chromosomes. Sometimes those pieces accidentally hook together. A chimp only has, you've got to think about this. I think the chimp is who has 24, and you and I have 23 because 2 of them appear to have gotten hooked together. 98%, it's a strange number since we used it already, 98% of your genome and mine are the same as the chimp has. How much? Would it be easy for an evolutionist to say, look at this. This is pretty good evidence that we came from. Are y'all with me on this? See. On the other hand, you could say, it's remarkable how the Creator is able to take just a handful of genes and make such a different creature. Y'all with me on that? Powerful stuff if you think about it. And I'd love to take time. I don't have it this evening. You don't have the time is the point. But it does look like we have one chromosome that looks like it came from 2. Because when you compare it to the 2 that the chimp has, you can see. There they are. They're almost the same. But they're hooked together for you and me. Anyway, that's the DNA. That thing, the artist has cut the corner out of it. He's cut the top off of this one. There's a whole one. These are the furnaces. These are the engines. When you put gasoline in your, I know this is a silly question, but please, it's an illustration. When you put gasoline in the tank, where does that gasoline end up? In the engine. It's the gas tank, I understood that. But after that, where does it end up? Talk to me? In the engine. And what happens to that gas in the engine? It gets burned up. And it provides energy for you to drive down the road. You all know that, of course. These blue things are the engines, where the food that you eat is burned for all the energy we have to do everything we do. No exception to this. No exception. By the way, when we eat food, we get fat, carbohydrate, protein, vitamins, minerals, and something called phytochemicals and something called fiber. Only 3 of those things are fuel: fats, carbohydrates, and proteins. And they are taken to this engine, if you wish, this furnace. It's like your house. Where does the natural gas go to warm your place? It goes into the furnace. This is the furnace where it burns the food that we eat and provides the energy for walking, talking, thinking, pumping, everything our bodies do. And this is an interesting one. Can you see the little black dot that's pointing to from where you're seated? Yes. That is a factory where proteins are manufactured. Is there just one in the cell? Hundreds, thousands. Now here's an interesting problem. The factory where proteins are manufactured is made of protein. Now you got a problem there, don't you? Which came first, the chicken or the egg? Y'all with me? We'll see some more of this tomorrow night. Now this is a picture, it's actually a photograph through a microscope of a cell. And the man who speaks about it is Michael Behe, whom tomorrow night you will hear this interview. So here he is.
At the very basis of life, where molecules and cells run the show, we've discovered machines, literally molecular machines. There are little molecular trucks that carry supplies from one end of the cell to the other. There are machines which capture the energy from sunlight and turn it into usable energy.
Now follow me closely. We have learned that for the average cell, and there are many different kinds of cells, brain cells, muscle cells, liver cells, pancreatic cells, on and on and on. We have learned that for the average cell, there's 100,000 different proteins in that cell that have to be there to make it work. Now, of any one of those, there might be 10 million or 400 million, but there's 100,000 different ones. Are y'all with me on this? And Michael Behe is saying they're little machines. There's machines that carry stuff. There's machines that are flagmen, and they say, they see a truck coming. You know what I mean by truck? One of the little machines carrying something that has to go somewhere. And this machine says, turn here. Now, there are genetic diseases just because the machine that said turn here doesn't work. Just because of that one thing, for example. So the complexity, folks, of the inner working of the cell is more than mind-boggling. I'm reading a book right now that goes down 50 more levels from what you and I are going to discuss here. It's taxing my mind. I couldn't repeat it. I can understand it while I'm reading it, but it is very challenging, what we know. And what we know is probably still scratching the surface. Everybody just realizes we're just barely getting in there. But it's enough inordinate complexity that's already known that people like Anthony Flew say, couldn't have happened. There is no way. And I'll finish this evening with an illustration for that. 100,000 proteins to make the cell work. Okay, there's all these different kinds of cells. And this is a picture of a female egg being under the attack of being fertilized. And the chromosome that is made when the sperm donates its half to the female half, the chromosomes that are formed, this is the one that came from the sperm, and this is the one that came from the egg. Are you all with me on that? And you've all seen pictures like this, haven't you, of chromosomes? Probably. This is what's going on. And they're fastened together. Interestingly enough, that's a PhD course to understand how that works. It's just incredible, folks, how much is known about the complexity and how much expertise there is out there among trained people on just little tiny pieces like this. This is an actual picture. After about 2 days, it takes about 12 to 24 hours for the first division to take place when that fertilized egg copies itself. And about 12 hours later, there's now 4 of them, and then the copying starts picking up. This is before the thing has been implanted in the uterus. We have different names for this. It's a trophoblast at this time, or just about, and then it's going to be implanted into the uterus. And we're going to look at this more in other evenings. I think you would know this term, don't you? And what that means is all of these cells are copies of each other. Exact copies, except for mistakes. And there's always mistakes made when DNA is copied. This may blow your minds, but it happens. But there are so many rungs that if 1 or 2 got off, it's still pretty much all the same. There are also stem cells. Any one of these cells can become anything in the body. You're aware of this, are you not? Any one of these cells has the information to become anything in your body. A brain, a heart with all the piping and all the electrical connections and all the valves. That cell has the information to make that whole thing. Or that cell has the information to make this whole thing. Or this whole thing. And I'll try to remember to do this. It's not in the slides. So I'm going to say it now and hope to say it again. You'll hear Dr. Carson make this comment. We no longer have to worry about getting stem cells, because we can take any cell in your body and make it back up to become one of these, and then by feeding it different stuff, we can make it become any organ in your body. So we can make stem cells out of any cell now. Did you follow that? Pretty fascinating stuff. Okay, we're going to close with this. We're going to make a protein together. And there's some things I'd like you to learn about proteins. After all, it takes 100,000 of them to make a cell work, right? Lots of each kind. And there's several things I want you to learn tonight about proteins. Number one, they work because of their shape. I shouldn't tell you this story. I'm going to tell you this story. When I was a young pastor, the couples came to me and they said, we want a couples retreat. I was worried about the singles, but we had a couples retreat. On Saturday night, the ladies had games to play. Be careful of those Saturday nights and those ladies' games. They put up a paper wall in a room about this big, about 9 feet high from that wall clear to the other wall. And they put all the ladies on one side. They put all the men on the other side with blindfolds. And they had the ladies stick their hands through some holes they had made in the paper wall. And the men were supposed to go down the line and do what? And guess who they had go through first? And I'm thinking to myself, this is crazy. Hands are hands, man. And besides, it's embarrassing. What if I end up with the wrong pair of hands? Now, about the fourth hand down there, I knew who that was. This gal was a farmer. And she has farmer's hand. Okay, I got that. But everybody else, I'm thinking, this is just crazy. But this is true, you ask her. I got down there somewhere, and I was feeling these hands. And I'm thinking, boy, those are familiar. And guess what? It was Neva. Proteins work because they fit. They work because of their shape. That's just a little illustration. I could tell by the shape, maybe the texture, whatever. You understand the illustration? Proteins work because of their shape. And they get their shape by connecting together the pieces or the building blocks that make proteins, which some of you will know, maybe most of you will know, that the building blocks that make proteins are called amino acids. How many didn't know that before tonight? Come on, raise your hands. You know it now, don't you? Not too complicated to simply know that a protein is made up of a chain of, and they hook together with something like a hydrogen bond, by the way. And what happens is, if we had enough people and time, I'd have you all stand up and make a big serpentine in here, back and forth through all the rows, holding each other's hands. And each person would represent an amino acid. And the whole chain of everybody would be a protein. And in fact, when the protein is first made, it's just a long chain. But then it gets folded up into these shapes. It's a very, listen folks, talk about integrated complexity. Because there are millions of different proteins, each of them working because of its, which is unique, and it's worse than that. This is your fault again. Here we go. It's not just the shape. It's the pluses and minuses that match the minuses and pluses on the protein that make it work. Is that elegant or not? The shape fits like a key, but it's more than like a key. It's electrical as well. Just absolutely. And you understand, folks, people start seeing this stuff and they say, whoa, how could this have ever just happened? You see? How could this have ever just happened? This is what Anthony, listen, you don't think Anthony Flew was a doomcough, do you? Huh? No. No. All right. How many different kinds of amino acids are there? 20. There's more than that, but we only use 20 kinds. In plants, this is where these things are found. And they have names. Don't even worry about the names. You know some of the names. You know that name? You can buy that in the store. It's supposed to be good for you. Don't waste your money. It's in everything you eat. Plenty of it. You know this one, don't you? Don't waste your money. It's in everything you eat. Forgive me, I'm supposed to stay away from nutrition during this series. Now, there they are. And don't worry about the pictures. Some of you have had a little chemistry, and you might say, oh, that's kind of interesting. They're pretty simple little things, these amino acids. Anyway, I'm going to pretend like the amino acid is a ball. That's what those yellow rings represent. Just think of the amino acid as a little ball, all right? Even though it's got these little parts. And let's hook a few of them together. How many amino acids are in this protein? 5. And it's a protein because they are hooked together. Are you all with me on this? All proteins are hooked together chains of amino acids, and they have names, and we get tired of using the whole names. So, by the way, I didn't tell you this, but I almost did, and I said I was going to wait a little bit. The average number of amino acids in a protein in our bodies is 200 to 300. Some 500, 600, 700, some 40, 50, 60, but the average is about 200 to 300. Depending on the need and how they need to be, how much there needs to be to have the right shape and the right electricity. And it has to be longer or shorter depending. So what the scientists do, instead of using names, they give all these things letters. Well, that's easy, A for that one and C for that one and so forth, but after a while, that's easy, G for that one, and over here you wished you could use a G again, but since you used it there, you got to use a different letter. Are y'all with me on that? So we just pick out 20 letters. And don't even worry about remembering them. Just understand that we use letters, okay. And so let's use letters instead of the names. Y'all with me still? And let's just shorten it up. Let's make a longer one just for the sake of interest. There's 10 of them here. This is just an illustration. I don't know of any proteins that are this short. Just an illustration. Here's the problem. If I make another protein and the only difference between number 1 and number 2 is the fact that the G and the L are in opposite orders, number 2 will have a different shape than number 1. So whatever number 1 was supposed to do, which it did because of its Some of you didn't say it. You all with me? Will number 2 be able to do that job? Wrong shape. Wrong shape. I wish I had time, because we do have evidences in some cases where there can be a mistake, especially if it's a large protein, and especially if the mistake is way down inside, where it would still work. Interestingly enough, just in case some of you have enough background in this thing to know about that kind of thing. Anyway, here's another example. Supposing instead of an S there was a Q, everything else the same, the shape of number 2 would be different than number 1, and it wouldn't do its job, see? So the plot thickens. This is a photograph in a microscope of some blood, and most everything you see there are red blood cells, and this is a white blood cell, so is this. You see they're not white, but don't worry about the color. Now the author is going to take this red blood cell and make it bigger and draw you a kind of a three-dimensional picture of it. And then he's going to try to take the tiniest point you could ever imagine and magnify it till you can see it, and that's a hemoglobin molecule. You all know the word, don't you? Most of you know that hemoglobin picks up oxygen from the lungs and carries it to everywhere in the body it might be needed, correct? I was with my mother when she died. I was holding her hand. She was unconscious. Maybe she knew I was there. And every once in a while, toward the end, she would take a short breath. They call it Kane-Stokes. A lot of you know about that. And when she took that little breath, her heart would start beating a little faster, and it would slowly slow down. And finally, when she took her last breath, in about 45 seconds, her heart stopped, because it was no longer getting enough oxygen. The muscles actually were not dead yet. The muscle fibers were still alive, but they didn't have enough ability with oxygen and energy to contract anymore. And hemoglobin, we all know this, is critical to life. There's almost nothing in our bodies, friends, that isn't critical to life. And it's all complex beyond imagination. So, just for fun, hemoglobin is a protein made up of 4 serpentines. You can see them, can't you? In case you don't follow what I'm saying, what the artist is representing is that these serpentines are made up of what? Amino acids, right? The little balls, okay? And it turns out that hemoglobin, in order to pick up oxygen and let it go, has these 4, but 2 are the same, and the other 2 are the same. These are iron atoms down in the middle. They're not protein. And it just happens that there's 574 amino acids in this hemoglobin molecule. And please don't be too afraid of seeing this. This is it. What do the letters represent? Amino acids. And these are the exact ones that make up the yellow chain and the blue chain. Y'all with me on this? Okay? Now, what I haven't told you is one more thing. I kind of said this, I did, but let's just make it clear. Every protein is built with the right amino acids because of a pattern of letters in the gene that tell the little factory which ones to hook together. Right? We're going to see it tomorrow night. Harvard University has made some beautiful video clips of this stuff happening. It's simulated, but it's very, very fascinating to watch. But just understand that all of these amino acids are in the order they're in because the gene has the information which amino acid to put next. Does that make sense? One after the other. In this case, 574 of them. And in some people, I'm just showing you that the yellow makes the yellow chain and the blue makes the blue chain. In some people, amino acid number 6 in the blue chain, instead of being glutamic acid, is valine. And I'm showing the pictures, don't want to scare you, but they're kind of alike. But just that one amino acid, the wrong one in the right place, makes the red blood cell, instead of looking normal, to get stiff and inflexible like that. Sickle cell disease. Is that amazing? This means, friends, that if you're going to get a protein and you're going to get it right, well, let me just show you. You kind of know this inherently, but I'm going to close actually with this little point about getting it right. How could it be? How could we form this by chance? How could natural selection do this? How could there be chance mutations and make even one protein correct? And here's the story. If you have a coin and you flip it and you want to get heads or tails, let's say you want to get heads. What's the chance that you flip it and get heads? You all inherently know it's 50%. I want you to understand why you know that. This will be important to see the rest of the picture. You know that because there's one chance out of two that you can get the thing you want. Is that correct? And you divide the 2 into the 1. What do you get when you divide 2 into 1? You get 0.5, don't you? Or you could change that to 50%. You understand that? Now you may not have known why you knew that, but that's why you knew that. You all with me on this? Supposing you wanted heads twice. What would be the chance when you flipped the coin of getting it heads twice the first time you flipped it twice? What's your intuition about that? 1 out of 4. 25%. Is that correct? Let me show you how you knew that. Well, you mostly didn't know it, but let me show you how you could have known it. You take the 1/2, which is the probability of getting heads once. Is that correct? Now watch this important point. You multiply it by the probability of getting heads again, which is still 1/2. You multiply those two together and you get what? Most of you know that you multiply the 2's to get 4 and you multiply the 1's to get... Is that correct? Is that how you multiply fractions? Yeah. If you've forgotten, that's how you do it. And if you divide 4 into 1, what do you get? 0.25 or 25%. Are y'all with me? That's how come you knew it was 25% chance of getting 2 heads in a row. Are y'all with me? Now follow me carefully. Have I lost you? Say no. Okay. What if I want to get all 4 in a row? I want to get heads 4 times in a row. 1/2 times 1/2 times 1/2 times, isn't that correct? You multiply, watch this, you multiply the probabilities together, okay? So that would be 1/16, or if you want 6%. Okay, that means if you flip the coin 4 times, you'd have a 6% chance of getting 4 heads in a row. Okay, now watch this. Well, let's forget the dice. It's the same idea, just to get on and finish. Let's take an amino acid, a protein that would just have these 5. Glycine, valine, glutamic acid, alanine, lysine. All right. What's the chance if I blindly reach into this huge vat that has billions of amino acids but the same amount of all 20? Y'all with me? What's the chance of me getting the right one, getting a G, if I just reach in there and pull one out? What's the chance? 1 in 20. And I divide 20 into 1 and I get 5%. Are y'all with me? What if I want to get a V on my next blind draw? It's still, for that one event, 5%. But to get the probability of getting them together, I've got to multiply what? I've got to multiply the 2 probabilities. So I've got now 1/400. Are y'all with me?
You lost me. Yeah. I don't do science. Keep going. Just keep going.
That's illegal. I thought you'd get the penny idea. 1/2? And you have to multiply the 1/2 times the 1/2 to get the probability of 2 in a row. Now my probability of getting heads is not heads. It's reaching in this vat and pulling out an amino acid. And how many different kinds are there? There's 20. So to get the right one, I got a probability of 1/20th. Y'all with me on that? And to get the next right one, I've got to multiply 1/20th times 1/20th. That's the probability of getting 2 of them in the right row. Got it? Not bad. Not bad. Okay. You know I'm teasing, of course. 0.00025. Listen, folks, this is just 2 amino acids in the right order. What about doing all 5? It's 1/20th times 1/20th times 1/20th times 1/20th times 1. All right? And the answer to that is 1 chance in 3 million of getting what? Just 5 in a row. Now let's go to a protein that's not nearly as big as hemoglobin. Let's take a smaller one to make it easy. And let's take a smaller one that only has 200 amino acids. Y'all with me? You got to reach into that vat, pull amino acid out, 1/20th, correct? Next one, times 1/20th. How many times? 200 times, and here's the number. 1 chance, and the way you say this number is you say the word 20, and then you say trillion 22 times. Listen, this is... Why isn't Obama here? Why in the world isn't he here? Now, this won't impress you like I would like it to. By the way, you can write this number in shorthand. There's the 2, and then you just say, give me 265 zeros. All right? That's just a shorthand way to write the number. Here's the shorthand way to write the number of particles in the universe. 100 billion galaxies. Every galaxy has about 200 billion stars. And those stars are made up of trillions and trillions and trillions and trillions of hydrogen atoms and so forth. Am I correct? This is how many particles there are in the whole universe. This number is way tinier, unimaginably tinier than the chance of getting even one protein that has only 200 amino acids getting it right. Y'all with me on this? This is why Anthony Flew said, this is one of the thousands of reasons why he would have said it's too complex. It could never have happened by chance. Now the people, friends who are committed to evolution, the Darwinian mechanism, you know what I mean by that. The people who are committed to that are starting to say in the literature, we know it's not possible by chance, but it happened because there it is. Now don't laugh at that, friends. If somebody has that opinion, I think I should respect it. Are y'all with me? I'm willing to have a dialogue. I'm interested to know their reasons. And I hope they're open enough to want to know mine. Are y'all with me on this? We're done. Good night.